Hi everyone, I hope you are doing great. Today we are starting a new topic about the T cell activation, differentiation and memory. Under this, we are going to discuss today about the immune synapse, which are the key structures in the activation of the T cells. So we will see their structure and their functions. So let's start today's class. Now, before moving ahead, let us see what we discussed in our last class. So we discussed about the B cell development niches, we discussed about the B cell development pathway, we discussed about the types of the B cells and B cell maturation, how B cells mature. All right. So that was our last class discussion. Now, if we see the initiating event which activate any adaptive response, that is when a naive T cell it interacts with the APCs, right? So naive T cells, they are just like the new soldiers who are coming out from the academy and they have been sent to the front. Okay, now they have never been exposed to any kind of antigen, so they when interact with APCs, that initiates the adaptive response. So here you can see in this picture how naive T cells and dendritic cell, which is a kind of the major APC, it is interacting, right? And once the interaction is successful, then T cells differentiate. Okay, now APCs with M.Sc presenting antigens on their surface, they settle in the T cell zones, and these T cell zones are found in the secondary lymphoid tissues, right? So APCs, they just they encounter with the antigens. When they encounter with the antigens, they take up the antigens and they present it with the MHC molecules on their surface. And when they present the MHC molecules with the antigen on the surface, then they settle in the T cell zones, and these T cell zones are in the secondary lymphoid tissues. So when they come there, right, they are scanned by naive CD8 positive and CD4 positive T cells. They keep scanning, and because they have the ability to recognize MHC class 1 and class 2, finding the appropriate antigen, they interact and then they stabilize the interaction. Okay, now, so if we see and look at the properties of the naive T cells before activation, right, they have not been activated yet, their chromatin is condensed. Okay, they have a very little cytoplasm, right, they exhibit little transcriptional activity, they recirculate continually, in blood, in lymph, in secondary lymphoid organs. So that circulation keeps happening. So one complete circulation takes them around 12 to 24 hours. Okay, now during this circulation cycle, if they don't find antigen, they again keeps recirculating, and that's how the possibility of finding the antigen increases. But if they find antigen, they stop and they activate, right? So here, once they find the antigen, once the APC interact with the T cells and the activation cycle starts, that is the initiating event of the adaptive immune system. Clear? Right. Now, let us see about the immune synapse, which are the contact point. So when T cell successfully interact with APC, they form an interface, and this is known as immune synapse. Now you must have learnt about the synapses in the neurology, right, where two neurons, they make contact and they form synaptic formation, yes, synaptic connections there. All right, now the same way, the same analogy has been taken, where in the immune system also we see such kind of connections there, and we call them as a immune synapse. So you can see here, this is the T lymphocyte, and here it is the APC, and you can see a number of membrane receptors, they are binding and they are making a successful connection and a stable connection here. Okay, so this immune synapse, it serves as a fundamental architectural framework for direct interactions and secretory crosstalks. So direct interactions, you can see here that the receptors are on both cells, and they are interacting, as well as the secretory crosstalk means the cytokines which are secreted by these cells, they also give the signals there. Now it can happen between either the immune cells when two immune cells are involved, or between immune cells and other type of cells here, right? So but when the dysregulation happens of this particular immune synapse, then this is thought to be an underlying cause of an immune evasion or inflammation, which you can see in various diseases like tumors and infection. Okay, so its regulation is highly critical. But if somehow dysregulation happens, then it culminates in different diseases including tumor and infections. The key regulatory mechanisms for immune synapse formation primarily include the polarization of the cellular membrane lipid rafts. Now you must have, I don't know whether you know this lipid rafts or not, but basically, just for the understanding, lipid rafts are the regions in in the cell membrane, and which keep floating, and they contain a number of receptors, right? So you know that the cellular membranes, they are fluidic in nature. Okay, and they have certain regions which are rich in these lipids, and they act like a rafts. Okay, just like the rafts, they float on the river, in the same manner these lipid rafts, they can float on the membrane, and they can house a number of receptors. So the polarization of the cellular membrane lipid rafts, so that they can interact with the other receptors on the surface of the other cell. Then the organization of the membrane proteins, how membrane proteins have organized themselves, that is also a key important decision making part here. The remodeling of the cytoskeleton, how cytoskeletons remodel themselves so that they can adjust and they can facilitate the interaction between these receptors, and the activation of the protein kinase signaling and dock. So protein kinases, as you know, these are the enzymes which are involved in the phosphorylation, and they act like a on and off switch. And usually, the ATP is involved here for the phosphorylation reactions. So protein kinases are very important for signaling, as well as the dock, and they are basically, the proteins which are helpful in the immunological responses, right? So if we see the feature of the immune synapses, then they have a bull's eye organization of the immune synapse. Now, bull's eye, you must have heard this term, and this is the bull's eye where the archers or the shooters, they aim, okay, to get the maximum points here. So you can see here they have the bull's eye organization, and when T cell binds an APC, okay, the contact site, it forms a highly organized and layered structure. Now how this organized and layered structure is formed, that we can see now. So let us see central supramolecular activation cluster, that is first in picture. So you can see here, this is the bull's like formation here, and in the center, this is the central supramolecular activation center. So this dark red color or maroon color, a ring you can see here. So it is located at the center. Of course, it is located here in the center, and it contains TCR. Okay, you can see here the TCR coming out from here, and that binds to the peptide M.Sc. You can see this TCR is interacting with the peptide M.Sc complex here, and it is a key signaling molecule. Okay, so it is the main site for signal integration and regulation. Remember, the central supramolecular activation cluster on the immune synapse, it is the main site for signal integration and regulation. Then the peripheral supramolecular activation cluster. Now it surrounds the center like a ring. So you can see here, this is the peripheral supramolecular activation center, and it is surrounding the central one like a ring here. It is rich in adhesion molecules like LFA1 on T cells and ICM1 on APC. So you can see here, this is LFA1 and this is ICM1 on the APCs here, and the function is to stabilize the T cell APC interaction. Okay, so their main function is to stabilize this interaction, and it keeps the cells tightly bound so signaling can persist. So basically it makes sure that the once the right signal is there, the signaling persists, okay, for the proper activation. Then the third is cluster is the distal supramolecular activation cluster. So you can see here, this is the distal supramolecular activation cluster, and this is the outermost ring. Of course, it is here. You can see here, it is the outermost ring. It contains cytoskeletal elements, especially F actin, and the function is to support the movement and organization of the receptors here. So you can see here the central supramolecular activation center, the peripheral supramolecular activation center, and the distal supramolecular activation center. It gives you a bull's eye appearance, right? Now, the second feature is the TCR microclustering. Now, this actually is the place where the signaling begins. Okay, so initial signaling, remember, it does not start in the central supramolecular activation center. Instead, the TCRs form small clusters. We call them as a microclusters, and that happens in the distal SMAC or distal supramolecular activation cluster, isn't it? So interesting. Now these are F actin dependent clusters, and you know that because they are the F actin dependent clusters, and they move inward, okay, centripetally towards the central supramolecular activation center. So basically actual signaling is taking place here in the distal SMAC, and then they are moving towards the center, and here that is the crux, right? Now third feature is that, and it's a properly a newer concept, that there is a exo central supramolecular activation center. Now what happens here, this particular complex is composed of vesicles which contain TCRs, okay, and these vesicles, they are released by the T cells and they are taken up by the APC. Okay, so here there are certain vesicles which contain TCRs which are released and they are taken up by the APC. This is a newer concept, and the function is to enable direct molecular exchange between these two cells, the APCs and the T cells here. It also enhances the communication between the T cell and the APC. Okay, now you know now that synapse is not just a contact zone, but it is also a bidirectional communication hub. Okay, so communication is happening to both the sides, and that is, you know, the key to the successful activation and differentiation of the T cells. Now fourth feature is that there is a bidirectional signaling between the integrins and the TCRs, and it is a two-way interaction, you know, between the antigen recognition and adhesion. So first is the inside out signaling, where from TCR to the integrins. So when TCR binds antigen, okay, then it activates integrins like LFA1 in this particular case, and that increases their binding strength. Okay, that increases their avidity. So there is a strong adhesion. Okay, then we have the outside in signaling, where the signaling is from the integrins to the T cells. When integrins bind their ligands, so integrins also bind their ligands, like LFA1 is binding to ICM1 on the APC, they send a co-stimulatory signals back into the T cell, okay, and that amplifies the activation signal, and that's how, you know, the activation signal is amplified. So that lowers the threshold which is needed for the activation. Therefore, this signaling is important. Now, let us see what are the key points here. The immune synapse is not just a structural, rather it is a dynamic and cooperative. Okay, so we have seen the special organization of these activation clusters in the form of central, peripheral and distal, and they ensure efficient signaling. Then we also have seen the microclusters which initiate and transport the signals here. And then we see the exocentral supramolecular activation cluster, a kind of complex which enables the molecular exchange. Okay, and then TCR and integrins, they work together to amplify the activation. So, in summary, the T cell activation depends not just on which molecules are present, but how they are spatially organized and interact dynamically. Okay, and that's how the T cell activation is not just a very simple process, rather, it involves a lot of signaling, a lot of interactions, and a lot of dynamic two-way the proper interactions between the cells and the ATPs. So now we are just closing our class today, and what we learned today was about the properties of the naive T cells and the structure and functions of the immune synapse. I hope you like this session, and now we will meet you in the next class. Till then, you take care and bye bye.